Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Modern medicine relies heavily on massive, complex biomolecules like proteins. But in nature, these essential building blocks do not exist in isolation. They are buried deep within dense, chaotic cellular fluid. To actually utilize these biological components for research or life-saving therapeutics, scientists have to perfectly isolate them from all that surrounding cellular noise.
If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated.
Traditional chemical separation methods like high-performance liquid chromatography, or HPLC, attempt to solve this with brute force. HPLC is a technique that uses immense mechanical pressure to rapidly force chemical mixtures through dense filters. The problem is that large protein structures are incredibly fragile. When you subject them to extreme mechanical pressure or harsh chemical solvents, they violently unfold and break apart, permanently destroying their biological activity. Researchers were caught in a bottleneck. The high-speed processing required for industrial purification was destroying the exact biological functions they were trying to harvest. In the 1980s, the industry developed Fast Protein Liquid Chromatography, or FPLC. FPLC is a liquid separation technique specifically engineered to operate at medium pressure, fast enough to be efficient for production, but gentle enough to keep delicate molecules intact. The system operates as a continuous automated loop.
Download our microbiology study app made for researchers like you.
Fluid reservoirs feed into medium pressure pumps, and a precise injection valve introduces the sample into the flow. The pumps mix water-based buffers to build a precise gradient, called the mobile phase, safely carrying the fragile sample through the system. This fluid gradient pushes the sample into the chromatography column. The column is a vertical glass or plastic tube packed tightly with a stationary phase made of microscopic porous resin beads. By replacing extreme mechanical force with a gentle, continuous fluid gradient, the system turns separation into a process of subtle physics. Inside the column, the mixture is separated based on the intrinsic physical traits of the proteins as they bump into the resin beads. In ion exchange, beads have specific electrical charges. Positively charged proteins stick to negatively charged beads, while neutral contaminants wash past. In size exclusion, beads feature microscopic pores. Small molecules tangle inside, while larger proteins bounce off and exit. To release proteins stuck to the beads, the system begins a phase called elution. Elution is the deliberate process of washing bound target molecules out of a column by altering the surrounding fluid. The system gradually changes the salt concentration, or the pH of the fluid gradient, forcing the proteins to detach at highly specific intervals. As the fluid exits the column, inline UV and conductivity detectors actively monitor the stream in real time. The moment they spot the newly isolated protein, they trigger an automated fraction collector to catch it safely in individual tubes.
If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated.
FPLC successfully isolates these targets by exploiting their invisible physical traits, like charge and size, pulling the mixture apart naturally without stressing the molecule's structural limits. Because the system intentionally operates at lower, safer pressures, a single pass through the machine rarely achieves the total purity required for medical use. Operators must run a time-consuming multi-stage workflow. They begin with an initial capture stage to isolate the target, move to an intermediate stage to strip away closely related contaminants, and finish with a polishing stage to reach final purity. This gentleness has heavy logistical costs. The process is slower and yields lower raw resolution than high-pressure systems. It also requires expensive, routine maintenance to prevent catastrophic clogs or biohazard leaks within the complex fluid lines. But that trade-off is exactly what allows the pharmaceutical industry to safely scale up the mass production of life-saving therapeutics. Recent hardware innovations are actively reducing those logistical costs. Miniaturized systems now save on expensive materials, while multi-column automated setups merge the purification steps into single continuous runs. Meanwhile, advanced UV detectors built with nanomaterials track the eluting molecules with intense precision. The system acts as a strict mediator in the laboratory. It negotiates the delicate balance between the harsh forces of fluid dynamics and the fragile complexity of biology, turning chaotic mixtures into pure, life-saving clarity.
Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!